Today, we will talk about the process of photosynthesis. And what we see in the background of this slide are plant cells. The plant cells are full of many small green dots that you can see throughout. These small green dots are chloroplasts, and chloroplasts are important because that's the location where photosynthesis will occur. Today, we will talk about the process of photosynthesis. And what you see in the background of this slide is a plant leaf with many individual cells, and each of these cells is filled with many small green dots. These small green dots are chloroplasts, and chloroplasts are the location for photosynthesis. As we will talk about, they are green because they are filled with chlorophyll, which is one of the most important pigments for photosynthesis. In our discussion of photosynthesis, we'll start by reviewing the formula, which we have gone over previously. We will then talk a little bit about chloroplasts, where they came from, and their structure. Next, we will talk about pigments, including chlorophyll A, which is the most important one, and accessory pigments, which also contribute to the process. Then we will talk about how plants use the energy captured from sunlight to store energy temporarily as ATP, and then they will then use that ATP to make glucose. Finally, we'll talk about the production of glucose itself, which occurs in the stroma of the chloroplast. As a reminder, photosynthesis is the process of acquiring energy, carbon, and electrons to build sugars, especially glucose. Remember, glucose is C6H12O6. The requirements for photosynthesis are: first, there needs to be sunlight or some other light source as an energy source. Second, there are some chemical ingredients. These include carbon and oxygen. These will both come from carbon dioxide, which has both, and this occurs as a gas in the air. Finally, photosynthesis requires a source of electrons, and these are going to get pulled off of water molecules. Some protons from water molecules will also be used in the process. We have here the general formula for photosynthesis. It is not balanced, but it has all of the different ingredients and products. So photosynthesis starts with carbon dioxide, or CO2, and water, or H2O, and when the process is complete, it will give us glucose, C6H12O6, as well as oxygen, or O2. And so this process then results in the production of glucose. Glucose, remember, is an organic molecule. That means it has multiple carbons as well as some other atoms. Carbon dioxide is an inorganic molecule. It has only one carbon, it does not have multiple, and so it is not complex enough to be considered organic. That means that in the process of photosynthesis, we are fixing inorganic carbon in the form of carbon dioxide into organic form in the form of glucose. This is what is meant by the term carbon fixation. And I also want you to notice that glucose is not the only product. As we already mentioned, oxygen is a product. From the perspective of the plant performing this, oxygen is really just a waste product. However, as you know, oxygen is also incredibly important for anything that respires, which includes plant organs, as well as you and I and all other animals. So this oxygen that's a waste product really enables animal life to occur. In the process of photosynthesis, carbon dioxide is going to be reduced. Remember, in the context of chemistry, reduced means that the molecule is going to gain electrons and sometimes protons too. So if we look here, carbon dioxide has fewer electrons associated with it than glucose, because glucose has gained all of these hydrogens and each of these hydrogens contains an electron. So carbon dioxide is reduced in this process. Remember that whenever one chemical gets reduced in an oxidation-reduction reaction, some other chemical must be oxidized. We'll look at that here. In the process of photosynthesis, water is the molecule that is oxidized. And you can see that because the oxygen in the water molecule here has hydrogens associated with it. But then once this process is complete, those electrons as well as protons have been removed, and all we are left with is the oxygen. There's two of them because two water molecules will combine together after this process has happened because the oxygen like to be paired. But neither of these oxygen has any hydrogens associated with it. That means it has lost electrons and it has been oxidized. That is a summary of the chemistry of what is going to happen overall in photosynthesis. We'll talk about where photosynthesis occurs, and it occurs in the chloroplast of plant cells. The chloroplast has various parts, and you can see black lines pointing to them. There are two membranes around them. The outer one is called the outer membrane. The inner one is called the inner membrane. Inside the inner membrane is where things get more interesting. First, there are flat circular discs of membrane, and each one of these discs is called a thylakoid, or you'll see them referred to as the thylakoid membrane. There is a kind of gel-like liquid outside of the thylakoid membrane stacks, and that gel-like liquid is called the stroma. And then there's a space inside the thylakoids that's largely an empty space, but it's going to be important for storing hydrogen ions. And that space is called the lumen. And I can rewrite, the word lumen is right here. So glucose will be made in the stroma. Hydrogen ions are stored in the lumen. Chloroplasts now are an organelle. They are one functional section of a plant cell. However, evolutionarily, they are derived from free-living photosynthetic bacteria. So we've mentioned that in addition to plants, there are bacteria that can photosynthesize. And it's really these bacteria invading some eukaryotic cells that created plants and hence created the ability of eukaryotes to photosynthesize. So this process of a bacteria being engulfed and then becoming part of a living eukaryotic cell is called endosymbiosis. And it's a big word, but we can break it down. Endo simply means inside, and symbiosis we've talked about before. This is a long-term close association of two organisms. In this case, the organism is going to be the eukaryote that made this eukaryotic cell, and the chloroplast itself, which was a free-living bacteria. So that is the structure within which photosynthesis occurs. We now are going to talk about the steps that have to happen for this process. And the first step is going to be that pigments absorb light. So what is a pigment? Pigments are simply molecules with light-absorbing properties. And the pigments involved in photosynthesis will be located on the thylakoid membrane. So these discs are the thylakoid, and the collections of pigments are sitting in groups on the surface of this membrane. These little lightning bolts indicate photons of light coming down and hitting these groups of pigments. We'll talk about a few different kinds of pigments that are important, but we'll start with the most important. The most important pigment in photosynthesis is chlorophyll A. And this pigment is special because it is the only one that is able to use energy from light in order to pull electrons off of water and then pass the electrons on somewhere else. And so pulling off electrons from water is super important because those electrons are going to be one of the building blocks for making glucose. The other kinds of pigments are able to capture energy, but they can't do this trick of pulling electrons off water. That means the only thing they can do is pass their energy on to chlorophyll A to allow chlorophyll A to do this trick. Here's a diagram of what chlorophyll A looks like. We're not going to worry too much about the specifics here. If I want you to notice something, let's focus on the details of what happens inside here. First, you can see that there is nitrogen, that's these 4 N's, and this is important because it lets us understand from the plant mineral nutrition chapter why it's important that plants have nitrogen. One use of that is that nitrogen is required in a chlorophyll A molecule. Second, you can see right in the middle of this magnesium, Mg, and let's just say it contains N and Mg for magnesium. This means that magnesium is another essential nutrient for plants. Generally, for pigments, the color of a pigment indicates the colors of light that it does not absorb. That seems backwards. We might think that chlorophyll, because it's green, is good at absorbing green, but that's not correct. Instead, it turns out that chlorophyll is able to absorb all colors except yellow and green. That means that chlorophyll is going to appear yellow-greenish to our eyes. Every other color is being absorbed by it, so the other wavelengths are not going to bounce off of it and end up going into our vision. What we see here is an extract of chlorophyll from plant leaves, and you can see that it's got a yellow-greenish color. You'll also notice this darker green color above it, and this is going to be a different form of chlorophyll, chlorophyll B. We'll talk about that when we talk about other accessory pigments. So accessory pigments is going to be our general term for any pigment that is involved in photosynthesis but is not itself able to pull electrons off of water molecules. Instead, they capture solar energy and they pass the energy only onto chlorophyll A, not electrons. So if these accessory pigments cannot actually remove electrons from water, we should ask, why are they present? It turns out that they are there because they are better at capturing wavelengths of light that are not well captured by chlorophyll A. Because accessory pigments are better at capturing other colors of light, this means that the plant is more able to use all of the colors of sunlight that hit the plant, and it will be more efficient. This would give it an advantage over other kinds of plants that had chlorophyll A only, but allowed other colors of light to pass through without being used. We've already looked at this picture, and I mentioned that there's 2 different colors of green here. One of these colors, this top one, is presumably chlorophyll B. And so you can see chlorophyll B has a different color. That means that it is capturing slightly different colors than is chlorophyll A. You can see it's only slightly different because both of these have a greenish hue. There are 2 other important groups of accessory pigments. The first was chlorophyll B. The other 2 groups are carotenes, which tend to be orange in color, and xanthophylls, which tend to be yellow in color. They are related, but they have slightly different chemical structures. So you've probably noticed, in fact I'm sure you've noticed, that leaves are green over the summer, and then many trees have leaves that turn either a orange or a yellow or sometimes a red color over the autumn before the leaves fall off. A common question is, why do leaves change color? And there are 2 explanations that are often given. One explanation is that leaves always have carotenes and xanthophylls. However, they have so much more chlorophyll that the chlorophyll covers up the color of the oranges and yellows. As a result, during the summer, the leaves look green. But then during the autumn, the plant starts to break down its chlorophyll so it can store that nitrogen for the next year. When that happens, then we see the carotenes and xanthophylls that were in the leaf the whole time. The second explanation for why leaves change color in the autumn is that the plants actually make xanthophylls and carotenes in the autumn for some reason. So these are both possibilities, and it turns out that we can actually test and see which one is happening. The way that we can do this is by extracting pigments from the leaves and then applying them to a special sheet of paper and letting the pigments separate. And when the pigments separate, then we'll be able to see the different colors associated with each pigment. And that will allow us to see, for example, whether there are carotenes and xanthophylls in a leaf that appears green, and conversely, it would also let us see whether there's any detectable chlorophyll in a leaf that appeared orange or yellow. The different color bands are going to be a clue to us about which different pigment types are present. And so this is an exercise that we will actually do in an upcoming laboratory. Thus far in the process of photosynthesis, really what we've talked about is light hitting pigments. And we've said that chlorophyll A can remove electrons. We now need to go through some steps that happen after light hits a pigment and electrons are removed from water. So the steps we just mentioned, pigments capture energy from sunlight, and sunlight is used then to pull electrons off of water, in the process oxidizing it. Now let's follow and see what happens. Those electrons are going to get passed to some neighboring proteins on the thylakoid membrane. And some of those neighboring proteins on this membrane have the capability of pumping protons. So the electrons, as they are passed, they activate proteins on the thylakoid, and these proteins are going to pump hydrogen ions into the lumen. Remember, the lumen was the space inside the thylakoid discs. Those electrons then, after they are used to do this, they don't go away. They still have to go somewhere. And it turns out that those electrons are going to get put on a carrier molecule. This is NADP. And that carrier, NADP, then is going to carry the electrons from the thylakoid into the stroma, and they're going to be used in the process of actually creating glucose in the Calvin cycle, and we'll talk about that process very briefly at the end of today's lecture. So we have now talked about how we get hydrogen ions accumulated inside the lumen. Remember that the hydrogen ion is simply H+ in notation, but this is the same thing as a proton. It's a single proton lacking any electrons. So really what we have are protons that are aggregated inside the lumen and many fewer of them on the loose outside in the stroma. This is going to be useful because this will function like a battery. If you have a battery, for example, in your cell phone, it will have two compartments, one with a relatively positive charge and one with a relatively negative charge, and it will allow electrons to flow from one to the other, and in the process that is going to release energy that can be used to power your device. Here a similar concept will be used. These hydrogen ions really don't like to be near each other because things with the same kind of charge repel. That means these hydrogen ions will want to leave, and as they leave, this is going to release energy. So by allowing a pathway for the hydrogen ions to leave, this is the equivalent of turning a battery on. We will see on the next slide that this battery will be used to make ATP. And so Here is a closer look at the pathway here that hydrogen ions are using to leave the lumen and go back into the stroma. Up close, what we can see, here's a hydrogen ion, and it is following this path out into the stroma. As this happens, it goes through a protein, and this entire protein structure up here is going to spin, and when it spins, it will take ADP and it's going to add this extra P onto it. This is a phosphate group, and that together will form ATP. Remember that ATP is our high energy form of storage, and ADP is the lower energy form. And so the energy that gets released by the hydrogen ion leaving is then used to power storing the energy on ATP. This will be important because the ATP will be used at the Calvin cycle that we mentioned previously when we actually make glucose. We are now up to the part of the lecture where we can talk about the Calvin cycle itself. The Calvin cycle is going to be the process of fixing carbon into glucose. And it will do this, remember, with carbon dioxide as well as water. And here's the list of ingredients. Carbon dioxide, the electrons that got pulled off of water way back on the thylakoid membrane that we discussed. And then we said they were eventually stored on a temporary storage, which was NADP, and then that was sent out to the stroma. Here it is now. NADP drops off those electrons, and they are added on. There are hydrogen ions in the stroma, and those are also used in the process of making glucose. The energy source for this entire process is the ATP that we just saw be made when those hydrogen ions left the lumen and came out to the stroma. And so together, we can take those carbon dioxides, put them together, add protons as well as electrons, and that will give us our C6H12O6. In other words, our glucose molecules. That completes our description of how glucose is made. The plant will typically take the glucose and then make it into some other molecule. For example, glucose can be made into starch for storage, it can be made into cellulose for structural support, or it can be made into sucrose to be sent somewhere else in the plant. It can also be used more indirectly as one ingredient to make a variety of other things like proteins, for example. So we're going to switch gears a little bit now. We talked about plant pigments earlier when we were talking about the initial stages in photosynthesis. We are going to now think a little bit about why humans and other animals benefit from eating plant pigments. Plant pigments have dietary benefits. This is because they can act as antioxidants. And antioxidants can help to prevent some aspects of aging and some aspects or some risk factors for other diseases. And antioxidants, what this means is that these are compounds that help stop unwanted oxidation reduction reactions. You already know that in oxidation reduction reactions, electrons are pulled off one molecule and put on another molecule. It turns out that sometimes in our cells, there are electrons that have more energy than we would like, and they start to cause a chain of oxidation reduction reactions. So electrons get taken from one molecule and put onto another one, and then that molecule puts them somewhere else, and that molecule puts them elsewhere again, and that process can happen over and over again. In this process of removing electrons and then putting them somewhere else, damage to various compounds in the cell can occur, and this damage then can make the cell non-functional or even eventually lead to its death. And so we want to have an ability to stop those chains of oxidation reduction reactions. Antioxidants are called this because they are able to stop these chains of oxidation reduction. So they can acquire these high energy electrons, but they're good at holding onto them rather than passing them on to some other compound. As a result, when the electrons get to the plant pigments we have consumed, then the chain stops and no further damage is done. One more specific use of plant pigments is carotenes, which we mentioned earlier. Carotenes, we said, have an orange color and are accessory pigments. It turns out that they are also vitamin precursors. Specifically, vitamin A is made from beta-carotene, and vitamin A is important in animals. In humans, it does a few different things. First, it's important to our vision. Second, it is important to immune system health, so it helps us fight off diseases. And it is also involved in a number of other aspects of growth and development. This means that eating an appropriate amount of vitamin A is one thing that we can do to help stay healthy. It is true that too much vitamin A, just like many other compounds, can have negative effects. So it's encouraged to have reasonable amounts of vitamin A, especially from natural food sources, but you want to be careful if you take supplements to not take more than the recommended daily allowance. Carotenes, and especially xanthophylls, which were another group of accessory pigments that we mentioned, are used, well, this isn't necessarily on purpose, but one effect of them is to give eggs as well as other dairy products like butter and cheese a yellow or orange color. This is most obvious in the yolk of an egg. This color is caused mostly by xanthophylls. This is also true for butter or for cheeses that have a very light yellow or orange color. Now, if you buy cheese that has a bright orange color, that is probably coming from a dye that has been added, not from the original milk that was used to make that cheese. The lesson then is that it is good for us to be eating plants like green leafy vegetables that have a lot of carotenes and xanthophylls for the antioxidant and vitamin precursors. We can also tell something about what animals that we eat have been eating based on the amount of yellow or orange in the products that they make. We will move on now, and we'll do a quick summary of the importance of photosynthesis, and then we'll end with some concluding thoughts. The importance of photosynthesis cannot be overstated. Photosynthesis is what allows animals to exist. First, photosynthesis allows plant growth. As we already talked about, it provides the glucose that allows starch and cellulose and other compounds to be made. Remember that all animals either eat plants directly, or at least all terrestrial animals, or they eat plants indirectly by eating other animals that eat plants. In other words, plants are the base of a terrestrial food chain. As humans, we are somewhere in that food chain. We both eat plants, and then at least most of us eat animals or animal products. Wherever we fall in that food chain, if we didn't have the base, then the rest of it would not be able to occur. Not only do we use plants for our food, but as we've seen, plants are the source of our oxygen. So we saw in photosynthesis that oxygen was a waste product. For plants, it's a waste product, at least from the perspective of photosynthesis. Remember that plants will be doing respiration, and of course, humans and all other animals are also performing the process of respiration. Oxygen is going to be required for this process, and photosynthesis is the pathway that allows this oxygen to be available to us. So without photosynthesis, then we would cease to be able to breathe. Tied to this is that photosynthesis is a process that removes carbon dioxide from the atmosphere. We will talk more about climate change and the greenhouse effect later in the semester. But generally, the more carbon dioxide that is in the atmosphere, the warmer the atmosphere is going to be. And so photosynthesis, by removing carbon dioxide, is a moderating effect on the climate. With that, we will conclude with a few final thoughts. First, remember back to the lecture about metabolism, and we saw in that lecture that photosynthesis and respiration are largely reverse processes from one another. We will be moving on to talk about respiration, and so if you understand photosynthesis well, it will give you a good background to understand what's going to happen in respiration. Next, I want to tie in what we've talked about with photosynthesis to what we talked about with water movement in plants. Remember that in photosynthesis, we need a supply of carbon dioxide. We also need to allow the oxygen gas, which is a waste product, to escape. And for both of these to happen, stomata, or the opening on plant leaves, must open. When the stomata open, this also allows transpiration to occur. Transpiration, remember, is water loss through openings in the plant. We will see that transpiration is important, first for moving water through the plant, but we will also look at, in a later exercise, the importance of transpiration for keeping temperatures cool, and we will look at this specifically in and around our homes. The thought that I want to end with is relating plant mineral nutrition to the process of photosynthesis. We've obviously seen in this process why it's necessary to have carbon dioxide as well as water. We also saw when we looked at the chlorophyll A molecule that nitrogen was required as well as magnesium as part of that molecule, and we specified that nitrogen is a component of proteins and enzymes are made out of protein. So for two reasons, we need nitrogen to be able to complete the process of photosynthesis. This means that if there's inadequate nitrogen in the soil, then there will be less photosynthesis. This is important both for understanding natural systems, and it helps us understand why fertilizing is important if we are trying to grow crops. As we move forward, we are going to switch from the process of photosynthesis, in which we build glucose, to the process of respiration, in which we break glucose down.